In chemiosmosis, we are starting with NADH. This electron carrier is, guess what? Carrying electrons. These electrons are released from NADH as well as hydrogen, resulting in NAD plus, electrons going down the electron transport chain, and hydrogen, protons, being pumped out to the other side of the membrane. FADH2 releases its hydrogens, resulting in FAD. Now, NAD plus and FAD are now able to circle back through glycolysis and the Krebs cycle. Electrons continue their journey to cytochrome B, cytochrome C1, and cytochrome C. The hydrogens released from FADH2 are pumped to the other side of the membrane by cytochrome BC1 complex. Continuing on with the electron travel, we see it arrive at its next destination, cytochrome A. From there, it moves to cytochrome A3, where it's used to form water molecules from hydrogens and oxygen in the cytoplasm of prokaryotes or the mitochondrial matrix of eukaryotes. Throughout this process, we have seen an accumulation of protons in the periplasm space of prokaryotes or the intermembrane space of eukaryotes. This is called a proton gradient. We also see an electrical charge gradient, which has a positive charge. This is called a proton motive force. Protons go through the ATP synthase, producing ATP from ADP and inorganic phosphate group. Chemiosmosis occurs at the mitochondrial membrane of eukaryotes and the plasma membrane of prokaryotes. Now, during aerobic respiration for eukaryotes, they produce 36 ATP, and prokaryotes produce 38 ATP. During the electron transport chain, eukaryotes produce 34 ATP, and prokaryotes produce 34 ATP. During glycolysis and the Krebs cycle, eukaryotes produce 2 ATPs, and the prokaryotes produce 4 ATPs. There is a low yield of ATP during anaerobic respiration. Now, of course, oxygen is not present, so the final electron acceptor is an inorganic substance. Here are some examples. The final electron acceptor for Pseudomonas and Bacillus is the nitrate ion. For disulfovibrio, the final electron acceptor is sulfate, and it forms sulfide. Another final electron acceptor is carbonate, and that forms methane. Let's define fermentation. 1, there is a release of energy from sugars or other organic molecules. 2, oxygen is not required, but fermentation can occur in its presence. 3, the Krebs cycle or the electron transport chain is not required during fermentation. And 4, an organic molecule is synthesized in the cell as the final electron acceptor. And 5, there is a low yield of ATP. Lactic acid fermentation. We have 1 oxidized glucose resulting in 2 pyruvic acids and the formation of 2 ATP molecules. Now, the 2 pyruvic acids are reduced by 2 NADH molecules, giving the end product of 2 lactic acid molecules, and this is where energy is stored. Here are 2 examples of homolactic bacteria, Streptococcus and Lactobacillus. They only produce lactic acid. Alcohol fermentation. We have 1 oxidized glucose resulting in 2 pyruvic acid molecules and the formation of 2 ATP molecules. The 2 pyruvic acids are converted to 2 acetaldehyde molecules and 2 carbon dioxides. The 2 acetaldehyde molecules are reduced by 2 NADH molecules, resulting in the end product of 2 ethanol molecules, and this is where the energy is stored. Here's an example of an ethanol producer, Saccharomyces yeast. Here are 2 examples of heterolactic bacteria, Clostridium and Salmonella. They produce lactic acid, other acids, and alcohols.